It's my pleasure now to introduce our second speaker of the day, Dr. Charles Danko. Dr. Danko approached science with a degree in biomedical engineering at the Johns Hopkins University, followed by a PhD at the Department of Pharmacology with Dr. Andy Persov at the State University of New York, Upstate Medical University, where he also worked as a software developer for the macroarray facility. He then moved to Cornell University in Ithaca for a joint postdoc between the groups of Dr. Adam Siepel and Kraus. And since 2014, he's leading his group at the Cornell University, where last year he was also appointed as an associate professor. We're really thrilled to have you here, and we look forward to your talk. The floor is yours.
Thank you very much, Matteo. That was a great introduction. And thanks to everyone for joining today. Can everyone see my slides okay?
Yes.
Very good. One never knows these days. I find I should be better at Zoom than I actually am. So I'm really an honor to be speaking to this community today. My lab has gotten a lot out of fragile nucleosomes since it was founded. It seems like a really great group of people who will be leaders in transcription and chromatin in the coming years. So that's wonderful. Keep doing it. Let me know if you need anything. So today I'm going to talk primarily about a preprint that my lab posted in July. And this is the work primarily of Gilad Barshad, who is a really outstanding postdoc in my group and is another person who would actually be a great to give the Fragile Nucleosome series. So this is again on bioRxiv. So check out the preprint and do let us know as you have questions and want to make comments. We're really eager to hear from the community about this work. So the story I'm going to describe today really begins with this fundamental idea that there's two types of regulatory activity. There are promoters which function to initiate transcription locally, and there are enhancers which serve to increase the rate of transcription from a distal regulatory site. So we've known about enhancers for 40 years or so since the discovery of this SV40 viral DNA sequence. And it was described as having this property that increased the rate of transcription from a target promoter without regard to its position or orientation, at least within a certain set of constraints. And this observation was fascinating because it implies that there's a mechanism of communication across long stretches of DNA. And really, it raises the fundamental question of precisely how this communication works at a mechanistic level. In other words, what is inside that fuzzy oval that you can see right here? So for many years, the dominant model of enhancer-promoter communication, I would argue, has been this enhancer-promoter looping interaction model. And so this slide shows a variant of this model, which was published by T.K. Kim and Ramin Shikatar in 2015. And I think it's quite representative of how many in the community sort of viewed the mechanistic basis of these interactions. So the basic tenets of this looping model is that there's this physical bridge between the enhancer and the promoter, which is filled in by protein-protein interactions that are speculated to involve mediator, perhaps cohesin, and other transcription-related proteins. But this elegant model was challenged by a number of really, I think, important observations that would appear at odds with these basic tenets of the looping model. So first, in 2017, Erez Aiden's lab put out this paper which argued that cohesin, showed pretty conclusively that cohesin degradation has a big effect on the high Cmaps that were described by Elias. But they have really only modest effects on transcription. Conversely, working with Rafael Casellas, Erez's lab put out a couple years later that mediator degradation has huge effects on transcription, but not much effect on a high Cmap. And then there's a variety of studies by a number of groups in the community which show that there is a physical separation between enhancers and promoters as they're undergoing transcriptional activation in both live cell and fixed imaging studies. So there's a bunch of these. Two of my favorites are this work by Thomas Greger's lab and Wendy Bickmore's lab. And in general, there's some variation in how far apart enhancers and promoters are observed to be, but it's in the range of 200 to 400 nanometers. So these observations have motivated the development of alternative models. So one that I think the community has discussed a lot in recent years is this hub model, or a condensate model. So it's the idea that has a somewhat older history that has sort of become an alternative. And it's early in this discussion, I would say. And there's still a lot of discussion about what this might actually look like. But the basic tenets of this model seem to be that there's a high local concentration of all sorts of transcription-related proteins, like transcription factors, coactivators, RNA polymerase. And these molecules tend to attract each other through weak and multivalent protein-protein interactions that together create this high local concentration that drives the process of transcription forward. So there's all sorts of speculation about this model and discussion about what it might look like. And I think that's really where we are as a community. So with these models as a backdrop, our work in this area over the past few years has been really motivated by reconciling these different models and specifically answering questions about whether enhancer-promoter pairs tend to interact, whether you can observe these interactions in high-C or micro-C data, what distance these interactions may take, and what's the sort of the ultrastructure, and then which proteins are involved in mediating these macromolecular complexes that lead to these interactions. And so that's sort of where I'll focus the talk today. So the technique we're going to use is micro-C, which is a lot like the high-C variants introduced by Matteo. So micro-C digests the genome down into mononucleosomes using micrococcal nuclease. It fills in the blunt ends using a biotin NTP, and then it ligates DNA in paired end sequence. And so it's a variant of high-C, and really I think the main difference is that micrococcal nuclease provides single nucleosome resolution of where there is an interaction junction. And I think this is an incredibly important innovation as we are looking at how these contacts relate to transcriptional activity. So here I'm showing two micro-C data sets prepared by Gilad in K562 and UrCat T cells, so two wacky karyotype myeloid or hematopoietic trans environments. So K562 and UrCat, and then below them you can see transcription, which is measured using a technique called ProSeq, which maps the location of RNA polymerase. And the data that I'm showing you contains two topological domains, one on the left here and one on the right here. And immediately you can see that in UrCat T cells on the right, this topological domain has higher transcription, whereas in K562, the topological domain on the left has higher transcription. And these differences in transcriptional activity correlate with the frequency of interaction of a lot of these enhancer-promoter communication across these two different cell types. So we know that transcription correlates with high micro-C signal. So the first question that we really set out to ask is whether there is regulatory information in these high-C or micro-C data sets in the first place. So not just taking all enhancer-promoter pairs, but trying to whittle down to those cases where the enhancer is actively functional in that it's driving transcriptional activity from the candidate target gene in that particular cell context or trans environment. And the way that we think about this problem is that if the part of the motivation for this is that if enhancer-promoter pairs really are farther apart, we might not expect to see that sort of function level information represented in micro-C or high-C data at all. Perhaps it's just a transcription compartment or some aspect of decondensation that leads to this correlation between transcription and high-C data. So the question we wanted to ask is there something specific about a functional enhancer-promoter interaction that leads it to have a higher residence time in this context of the nucleus? So as a window into this, we set out to define these enhancer-promoter pairs that were actually functional. And the way that we do this is we apply some sort of a functional perturbation in cells that can divide these regions into functional enhancer-promoter interactions where the enhancer is actively driving transcription at the target promoter, and those that are not. And really the advantage of this strategy is that it's an orthogonal source of information to the micro-C map, And it provides a way to control for chromatin accessibility, 1D genomic distance, transcriptional activity, that could confound your observations of contact frequency. So then among these groups, we can just simply ask whether there are features that distinguish these functional and non-functional interactions. And there's a variety of perturbation data in K562 cells from which we can draw. So on our first and now published foray into this area in 2019 in collaboration with John Liss, we focused on the transcription factor HSF1 and following the induction of heat stress. So in this system, there's a large number of genes which are activated robustly by heat stress, and a subset of those are activated by the transcription factor HSF1. We can take a plate of cells, we can raise the temperature to activate the heat stress, and we can do this in either wild type cells or cells that are cells from a knockout mouse or a siRNA knockdown of the HSF1 transcription factor. And we can divide these into two groups. For example, HSPH1 on the left here, you can see that there's a very rapid induction of transcription following heat stress. But in a HSF1 knockout mouse, this induction is completely abolished. There is no more induction following heat stress in this system. But for PT-PRM, you can see that there is nonetheless robust induction of the gene in both wild type and HSF1 knockout mice. And so we conclude that PT-PRM is not a target gene of HSF1, but HSPH1 is a target gene. So with this in mind, basically all we have to do to separate functional and non-functional interactions is ask if we can predict HSF1 target genes. So we've experimented with a variety of ways of doing this, and the best model was actually a fairly simple first principles approach that we called transcription factor dose. So in this model, we assume that there are two factors that are important for predicting whether a transcription factor, which binds to the genome, is actively regulating a target gene. And these are the activity of that transcription factor at a distal regulatory site and its frequency of interaction with the target promoter. So those are S1 and C1 respectively here. And this is a lot like the idea behind Jesse Ingrid's ABC model, if you're familiar with this. The main difference, I think, with Jesse's work is that we use this logistic function shown in the slide here to transform the high C contact frequency. And the reason that we do that is that we assume that at some point there's enough interaction for the enhancer to regulate the target gene, and then the activity of that enhancer becomes more important to determining whether that gene is going to be regulated or not. So that's the rationale. And this dose model performed better than alternative machine learning strategies, which sort of take into account the same kind of information. So here, dose is shown here, and then various machine learning models, which take into account all or a subset of this, is shown to the right of it. So this shows that there's really a significant amount of information. I'm sorry, I'm jumping around. I've lost my spot. In high C data, which is really exciting. But we'd also like to be able to say something about whether there is an increased residence time that enhancers and promoters, in other words, when they come together, do they come together more frequently, or do they spend more time together when they are functional compared to when they are not functional? So since we published that paper with John, there's been a number of new CRISPR-I experiments that have come out in K562 cells. And these provide, I think, a really nice window into knocking down the effect of an enhancer in this particular cell type. And so these are shown here, some of these references. And what these do is they knock down an enhancer and then look at the effects of that enhancer knockdown on gene expression genome-wide across hundreds of different enhancers. So using this strategy, we can collate a list of enhancer-promoter interactions where there's a functional effect and those where there's not a functional effect. There's no impact in so much as we can tell based on that data set. So in general, CRISPR-I defined in functional enhancers have a higher contact frequency in these aggregate peak analysis than non-functional interactions. And when you actually look at the difference that's shown on the bottom here in the red and blue aggregate peak analysis, the difference between the functional and the non-functional, you can see that there is a substantial increase in contact frequency, which could be interpreted as an increased residence time of interaction between the enhancer and the promoter. So the MYC locus is one great example that Jesse Ingrid's lab has dissected extensively. And in K562 cells, as in most other cell types, MYC has this huge regulatory window that spans about 2 megabases. And you can see the MYC gene over here on the left. It's just this small burst of transcriptional activity in the ProSeq data right here. And the rest of this window corresponds to regulatory elements that may or may not have an impact on MYC expression. And you can see that both by the divergent transcription in ProSeq, by divergent transcription initiation in ProCap, and then I'm not showing you other markers of enhancer promoter activity like H3K27 acetylation, but rest assured those are just about the same at this locus. They give just about the same window as the transcription initiation does. And we can categorize these as functional based on this CRISPR-I data from Jesse's lab, where these low values indicate a big impact of an sgRNA at that position on cell growth. And then the assumption is that that occurs through MYC expression, which I think they back up pretty nicely in this paper. So in this particular locus, these 7 enhancers, now I'm showing you virtual 4C data from, in this case, high-C data, where an anchor is placed at each one of the 7 different MYC enhancers in blue, and at intervening transcriptional regulatory sites in gray beneath that. And what you can see is that at these transcriptional regulatory sites, there's this increased signal close to the MYC promoter region that we can interpret as increased residence time. And you can quantify this and find that it's different at this particular locus. So it's nice to have that sort of locus as a case study. Finally, can micro-C shed light on any of the microarchitectural features of enhancer-promoter pairs? And specifically, can we get any information about the range of distances that might be permissible in this particular data set? So we would argue that there is. The argument is somewhat complicated, and I'll walk you through that now. So the idea is that if there's a transcription start site of a promoter or an enhancer, it sets up this phased array of nucleosomes that are sort of shown in the cartoon here. And you can see that if the distance is relatively small, that you can get cross-linking and ligation between these +1 nucleosomes that are very close to the transcription start site. So they can cross-link, and then the linker DNA can wrap around and ligate and give information in the high-C map. If the two are separated by a longer distance, though, the +1 nucleosomes are no longer close enough to cross-link and or ligate to each other. And you can still get signal from this region, but instead of having this be direct interactions between the +1 nucleosomes, these nucleosomes further along in this ordered array have to sort of swing around and interact with the nucleosomes between from the enhancer in this particular case. So note that this is what we actually usually are measuring when we count contact frequency in high-C and micro-C data, because we integrate signal across a relatively large window of 1 to 5 kb. So the question becomes, are +1 and +2 nucleosomes early in this ordered array overrepresented in the micro-C contacts? So genome-wide, if you look at aggregate peak analyses between here showing 20 base pair resolution, you can see that there is a lot of signal within this region. So here is the nucleosome-free region of the promoter and the nucleosome-free region of the enhancer in this aggregated peak analysis. And here's the interactions between the +1 nucleosome at the promoter and the +1 nucleosome at the enhancer. So there's a lot of signal here. And of course, this reflects the fact that the +1 nucleosome is really well positioned. And we're getting a lot of interactions between the promoter and enhancer here. at these well-positioned +1 nucleosomes. So we asked whether there's an excess signal in the functional contacts relative to the non-functional contacts between these early nucleosomes in the array. So of course, there's many less sites, so we can't show this as an aggregate peak analysis. But what we can do is we can take the first two nucleosomes from the enhancer and compare the contact frequency between those nucleosomes and the whole array at the promoter. And that's what I'm showing in this slide here. And what you can see is that there is a general increase in contact frequency, and that this peaks very close to the transcription start site of the enhancer. And the same plot is generated if you look at the enhancer and compare that to the nucleosome array at the promoter. Both directions give you the same plot. Is the important part. So the way that we interpret this data is that enhancers can get very close to promoters, and this proximity does seem to correlate with enhancer-promoter function. So I've shown you that there's a significant amount of interaction in micro-C data, that enhancer-promoter pairs can linger close together, and that they can come into very close contact, enough for the +1 nucleosomes to ligate. What are the proteins that facilitate this ligation? So we'd argue that an excellent candidate under most models of interaction is Pol2 itself. So in the context of the looping model, Pol2 could be a critical piece of this stable bridge. And for the hub model, there's several features of Pol2 that make it a very good candidate. And these are that the Pol2 C-terminal domain is an intrinsically disordered region that has been argued to form condensates, both in vitro and in vivo, and these are reported to depend on the phosphorylation state of the Pol2 C-terminal domain itself. And then there's multiple papers showing that RNA in a variety of different contexts can contribute to condensate formation as well. And most relevant for our particular case is this paper showing that M6A methylated RNA can form condensates that sort of have that can increase the transcriptional activity locally. So both of these suggest that the Pol2, which is tethered to the nascent RNA and the C-terminal domain, is a good candidate for being involved in these interactions. So because of this reasoning, it's somewhat surprising that the literature seems to be quite split on the extent to which Pol2 depletion affects contacts. And a great example is this really lovely study by the Darzak lab, where the authors used two small molecules to block transcription at different stages. So triptylide blocks new Pol2 initiation, and flavopiridol blocks release of a paused RNA polymerase from promoter proximal pause. So both of these empty Pol2 from the gene body and prevent new initiations, whereas flavopiridol leaves Pol2 in the pause position after blocking. So the authors use micro-C to examine what the impact of these drug treatments was on contacts. And what they found is shown in this slide. So they did find extensive evidence for changes. But what they found is that most of the changes happen in these regions that are away from the enhancer-promoter interaction, which they call the stripe. So compare, like, for example, this window here to this window here, or this window here for triptylide and flavopiridol. Whereas there were changes at the enhancer-promoter focus, which they call the dot, but it's just not as large as the change at the stripe. But I would argue that the challenge when interpreting these kinds of maps is that chromatin changes quite dramatically when you block transcription. And this change in chromatin has a big effect on MNase cut frequency near promoters and near enhancers. And we need to account for these changes when we're interpreting the contact frequency. So to adjust for this, Gilad developed two normalization schemes with the goal of removing the component of contact frequency that's driven by like just technical differences in micrococcal nuclease cutting. So these are shown here. I won't go into extensive detail, but in essence, the first normalization scheme looks at the ratio of direct enhancer-promoter interactions in a window around the enhancer and the promoter, and divides that by interactions between the enhancer or promoter and flanking regions around the other enhancer and promoter. And then the second normalization scheme builds a bulk aggregate peak analysis, and first it computes the matrix of the observed changes in the numerator between the two conditions. And then second, it computes the matrix of expected changes based on 1D signal across the anchors, and it divides these two quantiles, quantities to get an aggregate signal normalized for changes in cut frequency. So those are the two ideas, and I'll use them sort of interchangeably throughout the talk. They're slightly different ideas, and they're slightly different to one another, but they generally tend to agree with each other quite well. And the crux is that when we apply these two normalization schemes using that same data from the Darzak lab, what we find is that the biggest drop in contact frequency is this focus at the promoter and enhancer contact. This is the largest decrease. So I think what this analysis adds is pretty striking evidence that PAL2 really does seem to be directly involved in these enhancer-promoter interactions. And it's worth noting that these results are consistent with work from Akis Papontis's lab. Akis, if you're on, I apologize for the pronunciation of your name, using the oxandegron system as the perturbation for PAL2. So check this paper out if you haven't already. And then Gilad last night also reminded me to remind everybody that there's this paper on mediator as well. So we conclude that PAL2 has an effect. Can we show that this effect is related to either initiation or pause release? So recall that RNA polymerase initiates and pauses 20 to 60 nucleotides downstream of the transcription start site, and then is released into productive elongation. And one can show using mathematical arguments that the rate of initiation is correlated with the density of RNA polymerase in the gene body, and that pause release is correlated with the ratio of pause to gene body. So we can use this idea to isolate different steps in the transcription cycle. And in these APAs that I'm showing now, Gilad has split genes into quartiles based on their initiation rate here. And you can see that this explains the vast majority of the variation in contact frequency as shown by a variety of labs at this point. By contrast, PAL2 pause seems to have a small effect as well. But the interesting thing about the pause is that it's arguably more specific to the dot between the enhancer and the promoter. So here versus here, whereas the flanking sequence is pretty similar between these two plots. So that suggests that the pause might be involved. And Gilad has used an analysis to compare differences in transcription between K562 and URCat to sort of support this idea. The more pause you have, the more change in pause you have, I should say, for increases or decreases in gene expression between K562 and URCat, the bigger the magnitude of change in contact frequency in these two cell systems. In addition to that, if you compare the flavopyridol and triptolide data normalized as Gilad has, you actually see a larger effect in the triptolide, whereas in the flavopyridol, where paused RNA polymerase is still associated with the chromatin, you see a smaller effect of blocking transcription globally. And then to further this idea that PAL2 can facilitate contacts, we've turned to an oxandegron system. And the idea here is that the paused RNA polymerase is mediated in part by this protein complex called NELF, which we can degrade using the DTAG system. So this is work that was done by Abu Hassam in Kat Hadronkas's lab at MSKCC, where he stably tags both copies of NELF-B with an FKBP tag. And then after a few minutes of integration with DTAG13, incubation with DTAG13, you almost completely deplete PAL2 at globally. You completely deplete NELF-B globally. And this has a big effect on paused RNA polymerase by just 30 minutes of DTAG treatment. So Gilad did an experiment where he collected microC data following a time course of DTAG and then DTAG washout. And what you observe is that there's a small but really reproducible difference, a decrease in the amount of contacts between an enhancer and a promoter when you stably deplete RNA polymerase. That peaks at 60 minutes, at least in this time course. We don't know what would happen if we continued for longer, and then recovers as you wash DTAG out over 24 hours. So all of these experiments are really consistent with the idea that PAL2 is an important component in holding enhancer-promoter contacts together. And as a very brief aside, One recent result from our lab that jives with this is this one by Alex Shivu, where she examined the effects of Pol2 depletion on histone modifications. And what she found is that when she treated with triptolide for a time course of 4 hours, you lose H3K4 trimethylation and H3K27 acetylation at promoters and enhancers. And she can verify this using chromatin-bound histones in a Western format. And this jives very nicely with a paper from Ben Martin and his PhD advisor, Leanne Howe, showing that Pol2 depletion does, in fact, decrease these histone modifications. And the way we interpreted these results is that Pol2 is a key piece of a very big machine. And when you remove Pol2, the gears, which are no longer clunking into that Pol2 machinery, tend to just sort of fall apart. And it's sort of the same idea, I would potentially, one interpretation at least, is that it's the same idea as when you deplete Pol2, and you can see that this condensate, Pol2 is this key component holding the enhancer and the promoter into this, this hub, and depleting Pol2 seems to have a big effect. So I'm actually going to skip the PARP section unless people want to hear about it as questions, because I have gone over time. So thank you again for coming. Thanks especially to members of my lab and our funding sources and our various collaborators on this project. Yeah.
Wonderful talk, absolutely staggering, really nice, thank you. So we'll wait for the questions. I'll just take the first one. I was really impressed by when you show the interaction of the +1 nucleosome of the promoter and the enhancer, which is clearly where the strongest interaction is. Mm-hmm. However, if you start looking at the cross, it seems that the +1 of the promoter interacts more or less throughout the enhancer, while the +1 of the enhancer doesn't do the same with the promoter. Do you think that has any sort of meaning? So what I mean basically is that the vertical stripe is stronger than the horizontal one.
So you're talking about the +2 signal versus the +1 here?
No, I'm talking about the heat map on the top.
Oh, the heat map on the top.
Clearly,
yeah.
The strongest signal is there, but the interaction of the +1 in the promoter seems to be relatively strong across the enhancer, while the opposite is not true.
I see. So basically, this signal in this region is higher than this signal here, and you're asking about that. Yeah. So absolutely. My hunch is that that's primarily driven by that +1 at the promoter just being slightly better phased than the +1 at the enhancer. That is my best guess. Yeah.
Okay, so it's due to basically localization, not to some sort of sliding on one side, let's say, and not on the other.
It's possible that there's something really interesting like that going on. Yeah, I don't want to discount that actually, but okay. I mean, the simplest explanation.
Yeah, no, just curious whether you had any idea about that. Well, I also had a curiosity about the beginning of the talk when you distinguish functional into non-functional enhancers based on the perturbation that you do. How do you account for, how do you consider shadow or redundant enhancers?
Yeah, that's a great question. Basically, I think we don't. If there are two completely redundant enhancers such that we would not observe any change in gene expression, they will not come up. So absolutely, that is something that is going to be missed and a limitation of this analysis. Yeah.
Okay, and then probably Dan has a question.
Yeah, fantastic talk. I was wondering, when you were talking about Pol2 and its role in enhancer promoter connections, and you're kind of saying, two possibilities are the CTD or the RNA. Mm-hmm. Right? Do you have what are your thoughts on, is that RNA that's coming out of elongating Pol2 or RNA that's released from like a premature termination event?
Yeah, I mean, in principle, if this really is just this condensate that has lots and lots of multivalent interactions, both could play a role, I suppose. I mean, I'm really interested in this idea that perhaps Pol2 with the nascent RNA itself and with the C-terminal domain can kind of tether the promoter and enhancer region into some sort of an interaction. I think I showed a cartoon of what that might look like here. Right? So the idea is that these are engaged. A paused RNA polymerase can be, as you know well, really super stable. 1 minute to 10 minutes is sort of the range that's bantered about in the literature, which is an eternity in the life of a cell. And so one possibility is that this paused RNA polymerase or elongating RNA polymerase or whatever is sort of a stable feature in tethering the DNA to this regulatory hub. If you will, that's completely hand wavy, right? And but I'm fascinated by that idea.
And have you looked at all at like the distance from the TSS to the predominant pause site in terms of how much RNA is actually going to stick out of the pause polymerase?
That's a great question. I mean, as you know, it's 20 to 60 nucleotides, and the exit channel will protect about 20. So I think in terms of the RNA, it's these more distal pauses that could play that particular role. So, I mean, one of the motivations was this M6A methylation mark that is placed co-transcriptionally and I don't know if they know the timing, actually. I suspect it's there as RNA polymerase is paused. These sites are frequently at the 5' end of a transcript. Yeah, so yeah, lots of interesting questions to explore, I suppose.
Yes, thank you.
Yeah, I think Elias also has a question.
Yeah, thanks a lot. I was just wondering what your thought on the relationship with cohesin then, because in the paper you referenced that they talk about the interference between maybe loop extrusion and you can have transcription stalling. Do you think that could be part of the mechanism?
So loop extrusion, transcription stalling. Yeah, so I mean, I guess what I think is that loop extrusion and transcription are two very different and separate mechanisms that both contribute something to chromatin architecture and in different ways. I have been really, I guess, taken by this idea that cohesin is not necessarily a transcription complex, but maybe a molecular hairbrush, right? Solving this problem that you have this huge polymer. How do you prevent this thing from getting tangled up in ways that you can't then go on and untangle? So I feel like that's a very, potentially a very different function that maybe has some, I mean, evolution is going to use all sorts of different strategies, right? And you can imagine the edge of a topological domain forming a tether that sort of brings the distance closer. I don't know if that answers the question. I feel like I'm rambling now.
No, that's fine. Yeah, no,
okay.
Thanks.
Yeah.
Okay, then I think we are also past the hour. So thanks again, Charles, and also Elias for the fantastic talks. And we'll see you in 2 weeks for the next session.
Very good. Thanks again for the invitation, and great to meet you all.
Great to have you here.
Yeah, thank you so much.